All right. So we are talking about systematics of bacteriology, and the topic for today is numerical taxonomy. Right. So when it comes to numerical, the word itself is telling you there is something to be counted. Right? Numerology, numbers. So here is the original article from Burgess Manual of Systematics of Bacteriology, which is this from the book, right? It is, as you see, the editor for the particular topic is Peter Smith. He is one of the uh, early, what you can say, the people, researchers, scientists, I would say, who had contributed to the development of this method, numerical taxonomy, right? So as you can see, it was developed in 1950s, and the word says it is multivariate analysis. Multivariate, so variates, variations, multiple, right? So multivariate analysis, it is about multiple characteristics you are going to consider, and analysis is going to be done. Of course, you have to take help of computers. So in this particular sheet, we will just scroll through it till other people are joining it, and then we will come to the detailing about the concepts. So what we talk about is phylogenetic relationship of organisms. So it comes to relationship by ancestry or evolutionary relationship, right? That is what phylogeny is. Now when it comes to the methods to find out the relationship between organisms, you need to find out how connected they are, how related they are. And when it is the relation to be understood, you need to know what are the similarities between them or dissimilarities between them. All right. So based on these, you can cluster them or you can group them based on the similarities found and based on the dissimilarities found. And then you are going to go to find to the organisms which have certain similarities among the tested variants, and then you are going to cluster them, right? And the cluster could be at different taxonomical levels. Like here you can see taxospecies is the term, genospecies is a term, nomen species is a term, right? So taxospecies say a cluster of strains of high mutual phenetic similarity. Genospecies is the one, a group with strains capable of gene exchange. And nomen species is a group bearing a binomial name. Genus and species nomenclature system, right? So this is how you have different levels of clustering or different types of methods of clustering the organisms. For example, they have given here DNA hybridization or relatedness. If you are tapping, then you will be able to distinguish a genomospecies, a group of strains, by comparing DNA relatedness. But genomic similarity alone cannot be the only point that is going to be necessarily used to talk about genospecies, right? So genomic resemblance is included in phenetic resemblance. Genospecies is defined as which are able to exchange the gene, right? But here are certain terms which are new to you, and we'll be talking about all these terminologies. So it is genospecies, nomen species, genomospecies, taxospecies, phenetic characters, and so on and so forth. I'm just trying to connect you with whatever we have seen in the book so that you can correlate it during the discussion. So, how is the data collected for numerical taxonomy? What is OTU, that is operational taxonomic unit, and then how is the character defined, and so on and so forth. So, these highlights include certain terminologies what we have to learn in today's discussion. All right. So, numerical identification is to be done, and what are the basic concepts in it, and what are the limitations for the same. All right. So here I'm going to share the um, PPT, and then we'll come back to uh, the chapter again. Right. Yes. So when I say numeric taxonomy, you have taxometrisis. The word itself tells you taxo is for taxonomy and matrix. So matrices is a comparative of algorithms. That is what, or comparison of comparison done with the algorithms, or comparison that are using certain numbers and you are comparing them. So matrix is one term that you have learned in 8th 10th mathematics, right? Of course we are not going to go into the mathematics, but I'm just trying to correlate the terminologies. So 1950s it was talked about as multivariate analysis, and use of computers was taken help of for analyzing it. Now when it comes to numerical taxonomy, what is the aim? The aim is to divide a set of methods for classifying organisms. So we have different approaches. Till polyphasic we have been talking, but this is one of the earliest approach, and it has got certain advantages and limitations. We will learn all that. So numerical taxonomy is successful in most of its aim, particularly in defining homogeneous cluster. Homogeneous, the word itself tells you that there are similarities, and clusters are the groups of organisms which are put together based on the similarities or dissimilarities they share, what is common in them, right? So homogeneous cluster of strains. Of course, you can characterize these homogeneity, you can find out by comparing different characteristics, which could be either antigenic nature of bacteria, or physiological processes and parameters, or morphological appearance. All these characteristics can be compiled together, and then you are going to cluster the organisms. How? We will learn, right? So clustering the homogeneous ones into a group, that is what is a concern, right? Now is that all? Just give me a second. So is that all? But this is the important term to be remembered, right? Now what next comes is taxa are polythetic. That is another term. That is, they consist of a group of members which share high proportion of common attributes. When I say common attributes, that means you are comparing the different parameters, and then you are sharing what do common things are known and are to be taken into consideration, right? Now when I am going for taxa as a concept, you are going to get a term, that is taxospecies. So what is taxospecies? It is a cluster of strains which are having high mutual phonetic similarity. We will come to the term phonetic also, we will learn that also, right? So you are going to find out how closely these organisms, they resemble each other, what common properties do they share, and that is utilized in any type of taxonomy, but here you are going to talk about numerical expression of it. All right, so here you are going to compare certain invariant properties, and invariant properties, that means they are not going to change, are constant, which are going to define the group, right, without permitting any exception. So all are going to have that particular characteristics, and they are said to be demonstrated by each organism in that particular group. Now, when it comes to the term genomospecies, it is a group of strains that have high DNA relatedness. So, genomo is to genome, all right? So, this is how the terminologies they go. And of course, it should be emphasized that groups with high genomic similarity are not necessarily genospecies. Genomic resemblance is included in phonetic resemblance. Resemblance, similarities, right? So, when we talk about genospecies, it is defined by gene exchange, and genomo is to relatedness in DNA. These are the two different things to be remembered. Right. Now we go to the next slide. Right here, other points are there to be remembered. Operational taxonomic unit, OTU. So OTU can be species, it can be strain, it can be genus. So these are different levels in taxonomical hierarchy, we know, and each of these can be referred to as operational taxonomic unit. At different levels you are going to work. Strain related OTU, when you consider, it is the highest level of, you can say, classifying the organisms in terms of there are minute differences also are taken into consideration, and the organisms are separated into different strains. We know that. Now, for example, we take a characteristic, and that characteristic is going to be analyzed for particular group of bacteria, as in the practicals we are doing for lactic acid bacteria, right? Here, uh, a particular character is defined as a property, right? And this can vary between OTUs. So if I am talking about strains, then this OTU, that property will be varying in the within the members of the that particular species, and is made up of different strains. So each strain will have its variant response to the particular character that you have selected. What do I mean? Say for example, you are talking about whether the bacterial species that you are working with are having spores or no spores. Okay, you have selected those which are having spores. Now on the basis of that, you are further going into position of spore and the length of the spore. For example, you are talking about a character, that is length of the spore, and 1.5 11m is one of its states. What does it mean? We'll come to it. It is important to compare the same character in different organisms, right? And recognizing that character are the same is called determination of homology. So we, in the first slide, we have talked about homogeneous clustering, right? So you have to select one character and find out how different members of that particular species. If OTU is strain, that is operational taxonomic unit is strain. So different strains are there under a particular species. So you are comparing different strains and you are looking for that property, that is say presence of spores and then the length of the spore. And based on length of the spores, the isolates which are showing similar size will be clustered together, right? Now it's not the only character. You have to go on for many characters. More the number of characters, better the clustering would be, and good would be the assignment of OTUs, that is in the strain, whether it is same strain or whether it is a differing strain, that you will be able to compare only when you are going to do a thorough analysis with n number of different variables or different characteristics, right? A similarity value is to be calculated. Now, what is a similarity value? It should be thought of as an estimate of value that would be obtained if one could include a very large number of phenotypic features. So, it's not that key, okay, this is sporulating, this is sporulating, so this is similarity value 1 1. No, not at all. You are going to check n number of different characteristics of that particular isolate, and you are comparing it with same characteristics of other isolate, and based on say 60 characteristics, you are finding 42 similar and remaining are unsimilar. So based on that, you will be able to find out similarity value. How to calculate, we will talk. So this number of characteristics that you should compare should be more than 50. Minimum is said to be as important or required to say it is different from A is different from B. It cannot be just said based on only one parameter or 10 parameters. You should have good number of variation characteristics that are counted for variability or similarity, and the n should be 50 or more. Now when you are checking the similarity or dissimilarities, quality of data matters. That is what we are experiencing in the practical also, right? And we have seen what kind of problems you will come across when you are finding out the biochemical properties and responsive organism, and how the data obtained will not be consistent. Of course, in spite of taking all possible precautions and very stringent working, it has been found that microbial data are prone to more experimental errors than is commonly realized, and the average difference in replicate tests on the same strain is commonly about 5%. The same strain, same organism you are using, the same test you are running, but you are keeping 5 different replicates of that, and or 10 replicates of that, or 20 replicates of that, right? So when you are keeping the replicates, number of multiple tubes of the same test, same organism, yet you will find about 5% error or variation, right? So you have to out you work out in a manner so rigorously that the processes are standardized and the errors are almost nil or zero, which is not possible, but you have to attempt to reach to minimum error. Now come to the term phonetics, a systematic system of classification based entirely on degree of similarity or differences, that is phonetics. So some taxonomies thought that by taking enough characters, preferably more than 100, one will be able to come up with taxa that were clearly descendants of the nearest common ancestor, matlab lactic acid bacteria, taxon, taxa, and the operational taxonomic unit. That is what we are referring to, right? So here you have to go for assessing at least 100 of characters, and then you will be able to find out how these organisms are related and who is the ancestor for the a particular strain, because we know evolution continues and it can be convergent or divergent. It would the characteristics which are developed by organisms during the evolution process would be collectively uh, developing into a particular trait which is similar in many of these organisms. Initially, the organisms are different, classified into different groups, for example, and certain common traits select them into a particular trait which is going to put them in a one group, one cluster. So that is convergence, and divergence, that is starting from one, you are getting many of them. Exactly opposite terminologies. But okay, right now you can just remember phonetics is system of classification based entirely on degree of similarity or degree of dissimilarity or variability. Now we go to next term. Here, what we do with finding out the similarities and dissimilarities. We are going to take the count of these, and we are going to develop the graphical representation so that we can get to know how related these organisms are or how do they differ. So here are two terms. One is cladogram, another is phylogenetic tree. Right? Now, when I say a cladogram, it is a diagrammatic representation that shows relationship of closely related organisms. It is a type of a phylogenetic tree, but only shows relationship between clades with common ancestor. If they are related, then only you will get to know. So cladogram is a tree-like diagram which is drawn using lines. The nodes of cladogram represent splitting of two groups. So cladogram is representation. How it looks like tree. And the branching point or the node point, what you say, tells you about splitting of the two groups from a common ancestor. And the clades are built using molecular differences instead of morphological characteristics. However, cladogram can be constructed using correct morphological and behavioral data as well. So it's not that the only molecular differences you are going to look for when you are developing a cladogram. You have to take help of morphological as well as other behavioral and metabolic data also. Now come to another term, phylogenetic study and phylogenetic tree. When you say about different advanced molecular techniques, you can evaluate phylogenetic relationship between organism in relation to the evolutionary changes of the organism. And that is what you develop a diagram, that is phylogenetic tree. It shows you what? It shows you relationship between organisms based on their characteristics, their genetic background, evolutionary relationships, and so on. So how do I compare cladogram against phylogenetic tree? To compare that, a cladogram or phylogenetic tree, yes, uh, it is really very important to know how do they differ and what is the differences in the two. All right, so let's look into that. Cladogram is not evolutionary tree, right? So it does not tell you about the evolutionary relationship, but phylogenetic tree tells you about evolutionary relationship. Cladogram represents hypothesis about actual evolutionary history, hypothesized, right? But phylogenetic tree represents true evolutionary history based on what is the branching and what are the common things and how they diverge. You can think, you can put the theory about or hypothesis about evolution based on cladogram, but phylogenetic tree tells you how the evolution might have happened. All right, cladogram is drawn with equal length The length of the branch does not represent evolutionary distance. Now, what does it tell you? Branch and distance, these are the two words. So, you can imagine any simple tree, right? Say a mango tree you are thinking about. What do you have? You have a basic stem, then the branching points, big branches, and they split out into smaller and smaller and smaller branches. But all the big branches which originate from the stem, are they of same length? No. The distance of each um, branch is going to be different, or any leaf at a particular point from the start point is going to be different, right? So that is a distance. And branch is very much like the point at which it divides into two. So branch uh, is going to tell you that they are differing, they are separating at that particular point, and the distance would tell you how far they are related and how far they are different from each other. So evolutionary distance also is important point in this. Branch length of phylogenetic tree indicates evolutionary distance, but in case of cladogram, it does not describe the evolutionary distance. So cladogram does not indicate amount of evolutionary time, but you can get to know about evolutionary time based on phylogenetic tree construction. So these were the terminologies related to the numerical taxonomy and related terms. Now, how do we go for when we talk about classification and use of numerical taxonomy in that? What we do, we go on collecting the data, right? Then the data must be coded and scaled in an appropriate fashion. What is this coding that we will talk about? Then you are going to find out similarity or dissimilarity, and based on that you are going to calculate percent similarity, and you are going to develop similarity matrix based on the selected set of characteristics. Similarity matrix, similarly you can develop dissimilarity matrix also, right? Or for a given test, how many strains are showing it positive. If they are showing that test positive, they can be put together for that particular test. But two strains, they are showing uh, say for example, as today we observe weakly positive and strongly positive. So you have to compare between the positive response also, isn't it? So that positive, how strongly positive, how weakly positive, that also could be one of the parameters when you are going to find out similarity and are going to put the similarity matrix. The similarities are analyzed for taxonomic structures, and that would help you to get the information or group the organisms into clusters. Right? So the strains are arranged into phenons, that is phenetic groups. One of these strains, then you have species, then you have genus. That is how we go, right? So this is what is broadly evaluated with taxonomic groups, that is called as taxa, which is equated to taxa. The property of phenol can be tabulated. Now, what you have done, you have collected the data. The data is in the coded form. What kind of codes? Of course, there are different types of coding systems and algorithms and computerized systems also are available. But very simple one we will take and we will proceed with it. So you are going to find out the similarity. You are going to find out the similarity matrix, and you are going to analyze it, and based on the analysis you can develop the cladogram and or the phylogenetic tree and find out how the organisms are to be clustered together. So these are the general steps when you are talking about it. Now, when it comes to the methodology, let's go into little details. So selection of taxa, that is the first step in classification, right? So what operational taxonomic unit you are choosing from the uh, from the different levels like strain, species, genus. That is how you go on till kingdom, etc. Right? So these are different levels. So you have to choose the OTU, which may refer to individual organism or population or species or genera. That is first. Then selection of character. As we talked earlier, you should characterize the organism thoroughly, at least 50 onwards. Up to 100 characteristics may be considered when you are finding out the description about, or you are measuring the performance of the organism with reference to a particular characteristic. You have to have large number of characteristics that are analyzed. So it is very required. Of course, as some systems these days are computerized, it is easy to analyze once the data is given to computer, right? Now, construction of character taxon matrix, how it is done. Now, the coding of characters and character state is given in a tabular form, and it is called as character taxon matrix. In character taxon matrix, there are rows and columns, right? So rows represent the character and column represents the taxa. For example, here the example of plant is given so that you understand it easily. 5 plant species, they are to be classified. Suppose we have the following data in which one represents presence and another represents absence. So how we give the presence of complex leaves, for example. You give 1 if it is present, and if it is absent you are going to mark 0. With reference to previous example, that is in case of bacteria, presence of spore, absence of spore. Presence of spore you mark it as 1 and absence as 0. So 1010 is a unique system, you know, that is the system that is used in matrices and in computer algorithms, we know that. So you are going to give 1 for positive response and 0 for negative response, and that is how you are going to develop a table. So what you are going to have, the data is going to be character taxon. So rows are going to represent character and in column the taxa. So character, and whether it is 1010 coding initially you need to do, and then you have to measure the phonetic similarity. So the character state, that is presence absence of a character, are compared, and you get to know about what is overall similarity between the different organisms. And then you have to find out each character which is having similarity and the character which is showing dissimilarity. So each character is compared with every other character, right? So spore, ओके, प्रेजेंट एब्सेंट 10 हो गया, लेकिन स्पोर at the terminal end, spore at the center, another characteristic, again it is 0 1. So spore present and location of the present, these are the two different criterias, right? You have to compare these two, and then you have to develop the similarity matrix. All right, so all these characteristics you have to compare. Each character is going to be compared with other character. This is very important, right? And results are expressed in terms of percentage, and it is given in the form of table where 100% similarity represents identical taxa and 0% indicates no similarity at all, which is quite obvious, right? Now here, when we are talking about coding, so let's uh, understand about it. Coding of the results, when you are doing different test reactions and characteristics, you are marking it as positive or negative form. All right, that is what we commonly do. Now here we have talked about a unique system, common system, which contains entries like plus or minus. That is what we generally do, or it could be put in the term of 1 and 0, which is common and convenient for use for you analysis using computers or for computation. All right, so what you are going to have, you are going to have TOTUs and score for n characteristics. N मतलब 50 characters, 60 character, 100 character, जितने भी है, right? So you are going to get the characteristics. You are going to compare presence or absence 1 and 0, and then each character is compared with another one to find the percent similarity. So of course, all the columns are not going to get filled in the table, but some of the columns will show certain rows which are uh, empty, that is gaps are possible. Right, so uh, OTUs and number of characteristics that are important. Now here there is one more terminology. Okay, this is what we have talked about. Let's take an example. Okay, here is one example, raw matrix. just to understand the concept, raw matrix, and converting it into proportion similarity, right? Now what is this? It is A B C D. Now A B C D, they talk about say human, monkey, and fish and shark. That is how they have taken the example of. And the characteristics, different characteristics, instead of that they have put the numbers 1 2 3 4 5 6 7 8. Why these numbers are? Because they are just tallying only one characteristics, that is sequence of amino acid in myoglobin protein, राइट एक मायोग्लोबिन प्रोटीन में अमीनो एसिड का जो सीक्वेंस है उसमें पोजीशन 1, पोजीशन 2, पोजीशन 3, पोजीशन 4, लाइक दैट इन द एंटायर प्रोटीन। जितने भी अमीनो एसिड्स हैं, पोजीशन 1 पे कौन सा अमीनो एसिड है, पोजीशन 2 पे कौन सा अमीनो एसिड है, दैट इज व्हाट दे हैव मार्क्ड, राइट? फॉर एग्जांपल, हियर इट इज 1 is human, second is alligator, third is fish, tuna fish, and D is shark. And this is 0.1 2 3 4 like that amino acids. So here M, G, E, these letters you see, they are the first letters for the amino acid. Now what do you see here? Here, or in all other places it is absent, but in B, that is animal, that is alligator, M is there, methionine maybe. If you see the 10th amino acid, all are having valine. But if you see the 11th one, three of them have L, maybe lysine or leucine, and then the fourth one, that is shark, is differing because it is having some other amino acid which is denoted by letter N. Alright, so for every amino acid such kind of a data they have generated. This is for one protein. It could be one of the characteristic, and within that character, that is myoglobin protein, they have analyzed the sequence from different organisms. Can they find out the similarities? Yes, for every amino acid at every position from 0 to say 11 taken here. It is not that 11 amino acid long protein, it is a really big protein, but to say the example we have taken it up to 11. So out of these first 11 amino acids, how do they differ with reference to amino acid present at particular location? That chart they have developed. Now based on that, they are going to see ki, in this case, okay, three of them are similar and fourth one is differing, isn't it? So based on this you have to find out the similarity. This data is converted into the distance matrix. So distance matrix मध्ये कसं काढणार तुम्ही distance की out of 4, 3 are showing similarity and one is differing. So 1 out of 4, right, into percentage into 100. That is how it goes. So you are going to get these distances calculated, which are going to be the decimal in 0 point something values, right? So this is how you are going to calculate distance matrix. And then based on that, this distance matrix is represented in terms of unrooted network, and this is what is called as the phenogram. Now this is percent similarity, and this is whether it is starting from a same point, they are diverging. Right, so here they are comparing C and D, A and B, how far they are close to each other, are they linked at all at any one point. That is what is calculated. Now how it is done, to understand in a better way, let's watch a video and then we will continue further.
What exactly is a tetragram and how are we going to use it?
So this is the characteristic noted for so many organisms. Now this is clustering shown. Close ones are put to one cluster. A, B, C, D, E, F, G, H, like that you have. Just observe how they are putting them together and how the lines are drawn. And D and E are similar. E and H is having some similarity, but the distance is different. Just observe it carefully. Using a computerized system, how you can go. Just observe. We will repeat it again tomorrow. So you are going for constructing a dendrogram.